Hello, and welcome to Group 21-3's presentation of our poster discussing the effects of pH on the enzyme catalase and its effectiveness in different environmental conditions. In this study, our goal was to discover how environmental factors change enzymes to make them more or less effective. We studied how temperature, concentration, and pH all had a role in enzyme effectiveness. For this poster, we decided to focus on pH as our independent variable to be a measurement of our enzyme's success. We measured the success by our dependent variable, the volume of bubbles produced when catalase was added to hydrogen peroxide in a different pH conditions. For greater enzyme activity, we should see a greater volume of bubbles, but when catalase is at less optimal conditions, we expect to see less bubbles. This volume of bubbles is the indication of the reaction of hydrogen peroxide breaking down into water and oxygen. Catalase speeds up this process, resulting in a greater volume of bubbles. Previous studies have found that enzymes will function best at an optimal pH, therefore, we expected to have different levels of effectiveness at different pH levels, with one optimal pH level showing a greater volume of bubbles. As research has shown catalase to be most effective at a pH of 7, we hypothesize that the reaction activity will grow as pH levels rise to this point and then begin to drop off as our solutions leave the optimal pH range.
Basic background information for methods. Rule 1, we placed the test tube on the test rack. Second step, we then added 1 milliliter of catalyst to the test rack. Third step, with a disposable pen, we've added 4 milliliters of hydrogen peroxide. Fourth step, we then led us to add 2 milliliters of distilled water. Fifth step, we then overall stir the chemicals together and then place the rod we stirred in a disposable bin. Sixth step, then we measured the volume of the bubbles at a time of 20 seconds to get the results. After all these steps, you'll be able to see the results of the bubbles on each of the pH solutions, which is then implemented by the chemical we added. Table 1, pH volume chart. This table represents the overall of the experiment and shows the bubble volume and the pH levels of each of the phases. On the left side, we have phases 2 through 6, and in the middle, we have the pH levels through the phases, and on the right side, we have the volumes.
For our graph, we displayed the enzyme activity and bubble amount compared to the pH of our solution. Our graph shows that we had the most enzymic bubble activity at pH lab 7, with the enzyme being less effective at different pH levels. The results of this were the Y column axis shows the dependent variable, the volume of the bubbles in milliliters, and the X column axis shows the independent variable, the pH value. No reactions were observed in our control when catalyst was mixed with water without any hydrogen peroxide.
In conclusion, our experiment demonstrated that catalase has the highest activity at a neutral pH of 7. This aligns with our hypothesis, suggesting that the enzyme structure is most stable in a neutral environment. When the pH was either more acidic or more alkaline, the catalase activity dropped significantly, likely due to enzyme denutrition. These results highlight the importance of maintaining optimal pH levels in biological systems for enzyme function. Catalase in particular plays an important role in protecting cells from oxidative damage by breaking down hydrogen peroxide. Disruptions in pH, whether due to environmental factors or psychological imbalances, could reduce enzyme efficiency, impacting both health and ecological stability.
And with the limitations and assumptions, there were 5 limitations and 5 assumptions, with the limitations being optimal pH range, denaturation, buffer capacity, substrate availability, temperature effects. And with the 5 assumptions, it was single enzyme focus, uniform pH, linear relationships, steady state conditions, and physiological relevance.